So now let's get into eukaryotic cell division, which is way more complicated than prokaryotes because we have a lot more going on. We're much larger, we have all these different organelles that we have to worry about, and our genetic material is in a much larger multiple chromosome format. Now, with eukaryotic cells, because we have all these complicated things going on, we don't do everything at once. Only some of our time is spent dividing. So we're not going to be going through that 20-minute rush hour project that bacteria were going through. Instead, we're in a very tightly regulated process that is dependent on what we call the cell cycle. Now, previously we mentioned that there are two major cell types in eukaryotic organisms: somatic cells and gametes. Somatic cells being body cells like your skin and liver and kidney and lung cells, and gametes specifically being sperm and eggs. Each of these types of cells are going to divide in a different way because there's different end goals for each of them, although both of them will go through the cell cycle before we go through division. So let's look at that cell cycle and discuss what's going on there. There's 5 distinct phases which we can kind of lump together into some groups. There is the GAP1 or G1, S, and G2, which are all part of a more larger incorporated area called interphase. Then there's mitosis and cytokinesis, which many times are talked about together because they tend to happen together, although not always. In between these different distinct phases, we have checkpoints. And these checkpoints are like stop signs where we're going to check our work and make sure that the cell is in working proper condition before moving on to the next phase. If there's a malfunctioning checkpoint and it ignores the stop sign, that cell might go through without knowing that it's actually safe or logical to proceed. This is where we see things like cancer. So let's look at what's going on here. Let's start with GAP1 or the G1 phase of interphase. This is the primary growth phase here, and this is where the cell does its normal everyday tasks and activities. Sometimes we do go into G0, which is a resting phase outside of the cell cycle. Sometimes this can last for days, even years, right? But generally speaking, we're going to talk about GAP1. Think just normal day-to-day activities. Then we enter into the S phase. Think of S as standing for synthesis, because that's our primary goal in S phase, is to synthesize the new DNA. So DNA replication happens specifically in S phase of interphase. DNA replication does not happen during mitosis. So we've replicated the DNA, and then we go to GAP2. This is where we have a little bit more growth happening, when we're also preparing to divide. So we're making last minute checks, we're making sure we have all the proteins we need, the organelles are ready for division. Then we go into the mitotic phase. This is no longer interphase. The mitotic phase consists of 2 parts, mitosis and cytokinesis. Mitosis specifically talks about the nucleus dividing, so taking all those duplicated chromosomes that we replicated earlier on in interphase and separating them out so that each new daughter cell has a complete and identical set of DNA. Cytokinesis is the actual split, taking that cell and splitting it into 2, where we have 2 new nuclei, 1 in each of those daughter cells. But like I said, we have to make sure that this process is highly regulated. We don't want our cells just replicating on their own with no control whatsoever. There are a lot of things that can potentially go wrong, and we want to catch those before we go through to the next step. So let's look at 3 particular stop signs in the cell cycle. In the G1S checkpoint, this is where we're going from normal activities into DNA replication. And what we're going to do there is we're going to double check and make sure that the DNA has not been damaged, and we're going to make sure that the cell is actually ready. Do we have the nutrients? Do we have the supplies needed in order to actually go through division? If we meet those requirements, then we can pass through that checkpoint and replicate the DNA. In the G2M checkpoint, we're going to double check that our DNA has actually replicated properly. One of the things that can go wrong is that mistakes can be made when replicating DNA. Now, if we don't proofread that, those mistakes will get locked in. We do have the opportunity, or we do have the ability to go in and fix those mistakes as long as we have not passed this checkpoint yet. Think of it like when you're writing an essay. When you write an essay to turn in for school, you write the essay and then you proofread it. But if you hit submit before you proofread, any mistakes that you made, you're not going to be able to correct anymore. They're locked in. So we can think of the G2M checkpoint like that proofreading process you do before you hit submit. Our last checkpoint we're looking at is the spindle assembly checkpoint. This is going to happen in metaphase of mitosis. At this point, we're going to make sure that all of the chromosomes are properly attached to the spindle fibers so that when we go through anaphase, everything gets split just how it's supposed to be. Now, when it comes to replicating DNA, the process is pretty straightforward. DNA has what's called complementary base pair rules, meaning that base pairs only have one potential partner, its complement. And we see that A's always pair with T's and G's always pair with C's, and vice versa. That means that we can use one side of the DNA molecule, one strand, as a template for the other. We don't actually have to have both sides present to know exactly what the DNA molecule looks like. And that makes replication really, really easy. So what am I talking about here? Here's our DNA complementarity chart. Here's our parent DNA molecule. We say that's TAGCTA, and the other side is ATCGAT. We will split that molecule in half. So we split those two bases away from each other. Since A's always go with T's and C's always go with G's, we can use one strand to build the other. And that's what we see here. So TAGCTA, and if we go from the bottom, we see that our bottom A is pairing with T, our T here is pairing with A, our C here is pairing with G. And on the other side, we see that the A from the other side is pairing with T, this T is pairing with A, this C is pairing with G. And so if we look at the two molecules we get at the end, we see TAGCTA with a complementary strand of ATCGAT, and our molecule on the other side, TAGCTA and ATCGAT. 100% identical both to each other as well as to the original parent DNA molecule we started with. We call this process semi-conservative replication because one of the strands is always from the old original molecule and the other strand is always brand new. This also helps with the correction process because we can proofread the DNA to make sure the bases match. For the most part, we're really good at catching those, although sometimes they are not caught. This is a toss-up of whether or not that's good or bad, as the mother of all evolution is mutations, so we get cool new traits due to these small, unchecked changes. However, sometimes it can lead to harmful things like cancer.